Hello, my name is An Nguyen. In this video, I present about my first question. Question number 1 in chapter 8. One of the techniques used to create a huge number of identical DNA in vitro is polymerase chain reaction PCR. Start with a single DNA molecule, then creating billions of replications used to compare the nucleotide sequencing between two viruses. PCR have 3 steps. Step 1. Denaturation. Using heat 94 degrees to separate two strains by breaking hydrogen bond between base pair. Step 2. Priming. DNA polymerase. DNA nucleotide is the deoxyribonucleotide 3-phosphate A, D, G, and C. And DNA primer to add to target DNA. Due to a lot of primer, single string bind to primer, then to another string. The primer provides DNA polymerase with 3 hydroxyl groups. It's required for DNA synthesis, then cooling to 65 degrees to reform double-strand DNA. Step 3. Extension. Raise temperature to 72 degrees and rate of DNA polymerase replicate its string to produce more DNA. After repeating this step many times, the number of DNA molecules increase billions. Hello, my name is An Nguyen. In this video, I present about my second question. After study chapter 13, I believe virus, viral prions, and bacteria have similar and different points. They are similar because they can cause disease. However, some bacteria can help human body. For example, colonic microbiota have to synthesize vitamin K. Virus need to complete before injecting into heart cells. Complete virus called virions. Virus have nucleic acids, DNA or RNA, protein capsid cell, and they are possible to have bilayer phospholipid membrane envelope. Virus inject on organism, bacteria, animal, plant, human. Viroids have only circular single-strand RNA. They block protein synthesis in plant by binding to RNA. Luckily, they only inject plant, not humans. Prions are injected protein. They are only protein misformed prions, BRB with beta sheets. It looks like zigzag-like. Prion inject animal, human, and can cause cell die. Bacteria is unicellular, single cell. Eukaryotes have DNA, ribosome, and nucleus. Bacteria inject on organism and other bacteria. It may be normal flora, not cause disease, or pathogens cause disease. However, virus, viral prions need host for DNA replication for survival. They are considered to be not alive. Bacteria do not need host to survive. They are considered to alive. Hello, my name is Anne. In this video, I will present about my third question. After studying chapter 9, I believe heat would be more effective against microbes. Moist heat cause denature protein and destroy cytoplasmic membrane. Cell will die. Moist heat is more effective to control microbes than dry heat because water is better to conduct heat than air. 3 methods I recommend are boiling, autoclave, and use high temperature sterilization. Boiling time is important to realize at higher elevation water boil at lower temperature because atmospheric pressure is lower. Boiling does not destroy protozoa cysts, bacterial endospore, and some virus at sea level. I believe boil can kill gram-positive bacteria because boiling can kill vegetative cell of bacteria, most virus, fungi, and protozoa. Autoclave require higher temperature than boiling water. To require temperature, pressure is applied to avoid heat escape during boiling. Sterilization is steam with water boil increase as pressure increase at that temperature. Autoclaves kill bacteria, endospore, and on organism, so it also can kill gram-positive bacteria. Ultra-high temperature sterilization used to process dairy food, example dairy creamers. Liquid passed through superhot steam at 140 degrees for 1 to 3 seconds, then rapidly cooling it. This method can kill gram-positive bacteria and endospore because their temperature is over 100 degrees. Hello, my name is An Nguyen. In this video, I present about question number 4. I found the answer in chapter 10. We start with inhibiting cell wall synthesis. Cell wall make up peptidoglycans. Peptidoglycans make up protein sugar complex NAG NAM cross-linked by peptide chains. Cell make new peptidoglycans by inserting new NAG NAM subunits. Antibacterial agent block the cross-linked NAM. Beta-lactams is antimicrobial drug or functional group called beta-lactam ring such as penicillins. Beta-lactam block peptidoglycans formation, break up cement layer, cell wall become weak and less resistant to osmotic pressure effects. Water move into bacteria and cause bacteriolysis. Inhibiting protein synthesis. Protein plays an important role in metabolic enzyme. Pump and channel have to move material in and out of cell membrane. Cell use RNA to translate protein. Many antibacterial drugs could selective target translation without affect eukaryotes. It means the drug would not be harmful to humans because we are eukaryotes. For example, tetracyclines prevent tRNA by a site, so it does not allow to add amino acid into polypeptide. Inhibiting metabolic pathways. Metabolism is on chemical reaction in organism. If different metabolic hosts and pathogens, anti-metabolic agent can be effective. Protease inhibitor block enzyme which SIV need in replication cycle, so it cannot infect the next cell. It means HIV cannot make more virion. Encrypting cell membrane. Some antibacterial drugs disrupt the cytoplasmic membrane by creating a channel through it and cause damage to integrity of cell membrane. Amphotericin B is fungicidal due to attack to arosterol, disrupting membranes, cause cell lysis. Because cholesterol is similar to arosterol, human also is susceptible to amphotericin B. However, most bacteria lack sterols, so they are not easy to kill. Hello, my name is An Nguyen. In this video, I present question number 5. I found the answer in lecture 16. How organism cause disease part 1. Virulence is the risk of pathogenicity. That means virulence is involving ability of pathogen to infect a host and cause disease. Pathogens have traits, interact and enter the host, and have the ability to hide the immune system. The trait called virulent factor. Extracellular enzyme. Many pathogens secrete enzyme to dissolve structure chemical and avoid defense system. Hyaluronidase and collagenase degrade specific molecule. They work together to enable bacteria to invade deeper tissue. This is where hyaluronic acid in. This is collagen layer. Hyaluronidase digest hyaluronic acids. Hyaluronic acids is a glue hold cell together. Collagenase break down collagen. Coagulase activate locked-in protein, providing a hiding place for bacteria in lock. Kinases digest blood block, allow bacteria to invade damaged tissue. Toxins, a harmful chemical, can cause damage in immune response. Toxemia means toxin in blood, and it carries beyond the site of infection. Two types of toxin are exotoxin and enterotoxins. Exotoxin released by microorganism. By live microorganism, it destroys host cell. The source comes from gram-positive and gram-negative bacteria. For human, the body responds by producing antitoxins, prevent binding to the target. Exotoxins affect the gastrointestinal tract. Endotoxin can be secreted when gram-negative bacteria die or digest by white blood cell. It's come from lipid 8, outer line lipopolysaccharide membrane, LPS. Endotoxins are dangerous because it can cause fever, diarrhea, and inflammation.